So I have a tendency to shoot my mouth off when I shouldn't, and that really is the sort of start of this lecture. So in 2000, at a small collection of biogerontologists with Natalie Ann Gier, reporter from the New York Times, in the room, she asked the provocative question, when will the first person live to be 150 years old? And everybody was unwilling to actually say anything about that, except me. And what I said to her is that I think the first person to live to be 150 years old is already alive. And so that was reported by her in the New York Times, and a good friend of mine, Jay Olshansky, phoned me up, and he said, "You didn't really say that. You were misquoted, right?" I said, "No, Jay, I believe that very much." So he contacted Natalie Ann Gier and said, basically, Austad is out of his mind. Are you kidding? And furthermore, he said to me, "You're out of your mind, but I've got a book coming out shortly. Would you like to make a wager?" And so this is what we wagered. We each put in $150 into a pot, and we were going to wait 150 years. And in 150 years, at the rate of the historical rate of the stock market, this is probably worth about $11.50 right now. What happens is if someone has lived to be 150 years, as will certainly be true, then in the best case, I get all the accumulated money, or my family, my descendants get the accumulated money. And if no one has lived that long, then Jay's descendants get the money. Now, why did we pick 150 years rather than 140 or 130? There are a number of reasons, and I think it's important to do this before I jump into Darwin's impact on research and aging. Why 150 years? Well, first of all, it was substantially longer than anyone had ever lived. Some of you may not recognize this person here, but if you take a picture of her some 82 years after this, she looks like this. This is Jean Calment. This is the oldest woman that ever lived that we have good birth records from. She lived to be about 122 and a half years. And that was about 12 years longer than the oldest person alive, known to be alive in the year 1900 had been. So if you assume that we'll continue at that rate of progress, then a 150-year-old person basically wouldn't arrive until the year 2200, which is 50 years after our bet. The idea of this bet was that we wanted to reach an age that no one was likely to achieve unless there'd been a major breakthrough in understanding the biology of aging, and that we'd actually been successful at slowing the rate of human aging, which, even though most of us in the field don't talk about it very much, that's really the ultimate goal of our research. We don't care how long they live, actually. What we really want to do is increase and preserve humans' health for longer than it is. Another reason is that from an extrapolation of the oldest old in Sweden, you can see this is the oldest old, the average of the oldest old in Sweden from about the middle of the 19th century up until the year 2000. At that extrapolation, the estimated date of the first 150-year-old was in the middle of the 24th century. So all of this suggests that just the standard medical advances that we've been undergoing for the last 100 years ago are not likely to do it. Now, to win my bet, as I will surely do, only one person has to live to be 150. I'm not saying that this is going to be the human life expectancy. Only a single person does. That's 21% longer than the oldest person has lived. However, I'm perfectly open to the possibility that we might discover something in which 150 years becomes a not exceptionally novel age. And if that's the case, then it's hard to describe its impact on human society, family relations, almost anything you could imagine. The way I like to think of the benefits of this is, what if people sort of rarely, but not too uncommonly, live to be 150 years. What kind of dinner party could you put together? So I put together a kind of my dream dinner party. I mean, you know, you might have to go to the old folks' home and have the dinner party. So who are these various people? Well, there's the inventor of basketball. There's a famous journalist, publisher, a Nobel Prize-winning author, the Paris Hilton of her day, with the exception that Annie Oakley could at least do one thing exceptionally well. Another double Nobel Prize winner. In fact, this would be a great dinner party. And if we lived that long, we could have, you know, you could assemble people like this regularly. Now let's jump into the next part of this, which is how are we going to get there? And more particularly, what does Darwin have to do with it? Because I know you've been having this series on Darwin, and I really would like to really talk in detail about some of the impacts. So 70 days from now, exactly 70 days from now, February 12th, 2009, it's the bicentenary of one of the most important men in the last 200 years. And it's not Abraham Lincoln, who happened to be born on the same day as Charles Darwin. It's Charles Darwin. Now, you could always say that Charles Darwin had an enormous impact on the biology of aging because he had an enormous impact on every aspect of biology. But there's at least 4 things that he really contributed, although indirectly, because you may not know this, but there's currently a project to put all of Darwin's 14,500 surviving letters on the web, and about 5,000 of them are up already. And I've looked through all of them to see what did Darwin have to say about aging. In fact, it's one of the few things he never really had much to say except to complain about aches and pains. And you know, somebody wrote him about the fact that he was getting prematurely gray hair, and his father had gotten prematurely gray hair. But he never actually said much about aging. But that didn't mean that he didn't contribute. So what did he contribute? Well, the first thing is what he really contributed to biology, which was an understanding that there weren't just species, there wasn't the rat, the mouse, the horse, the person, but that there was individual variation. There were individual rats and mice and horses and people, and that genetic variation formed the raw material for evolution. But for us, it could also form the raw material for understanding how to age more slowly. And to give you some idea about the human variation for aging quickly or aging slowly, I'd ask you to guess the ages of these people. So who's the youngest? Who's the oldest? It turns out 2 of these people are related. Here are their actual ages. This Italian woman has lived a life out in the sun. It's done certain things to her skin. Bush woman from the Kalahari Desert that looks quite youthful at 50, at 68. She didn't look so youthful. And then this father-son pair. Now, these 2 come from a study by Nir Barzilai studying healthy centenarians. That is, not just people who live to be 100, but people who are healthy when they get to be 100. And I think you'll note that his 70-year-old son looks remarkably younger than 70. Clearly, there are some people who are aging in a different way than other people. And it could be, now that we have the sophisticated genomic technology, that we're ultimately going to understand the mechanisms that allow people to age so exceptionally well. I'll tell you another exceptional ager is Dana Torres from the last Olympics. Notice that she set a world record in the 50-meter swim, 25.69 seconds at age 26, and 15 years later set an American record that was over 1.5 seconds faster. Now, for those of you who know Olympic swimming, it's a young person's sport. This is the average age of people that won the 100-meter swim or the 50-meter swim since women were first allowed to compete in the Olympics in 1912. And this is the decline in the time for the 100 meters and the 50 meters. And this is what Dana Torres was here talk about an outlier, and her times at 41 years of age were here. So this is an outlier in a different sense, an outlier that's preserved exceptional health at least up to early middle age. So what can we learn from that father and son? What can we learn from her? That sort of variation is the kind of variation that really interested Charles Darwin. The second thing that Darwin contributed, again, indirectly, is he basically provided the intellectual framework to develop evolutionary senescence theory, which is really the best general theory we have for why we age at all. I mean, we wouldn't have to, right? This is a paradox to a certain extent. If you think about any biological organism, it can almost be defined by its ability to repair itself. But what is aging, if anything, as the ultimate failure of repair? So things wouldn't have to age necessarily. There's no principle of physics that we know that means things have to age, but they do. Now, like I say, this was Darwin by Haldane, Medawar, Williams, and Hamilton. I'd like to just talk a little bit about this theory, because on the way up here, I actually looked through Bob Cohn's book. Excellent book on the principles of mammalian aging, because I wanted to see what he said about evolution, the evolution of aging. In fact, he said nothing about it, even though these theories were around. And to me, that just indicated that there were two cultures. There was sort of the medical culture for studying aging. There was the evolutionary biologists who were interested in understanding how aging came about, why it happened quickly, some animals, slowly, in other animals. And for the longest time, those communities just didn't talk. You know, there's a role for an ambassador or for a spy from each of those to visit the communities and see what's going on, because you never know when you might learn something from somebody from the other community. And I have to say that that's changed. In the last few years, there are now plenty of evolutionary biologists that mix with the medical biogerontologists, and we really are starting to make serious progress because of that. Now, what's that theory? Well, what started off the theory was a simple conversation between two of those people, J.B.S. Haldane, one of my favorite characters of all time in biology. His dad, who was a mines inspector, used to test the air in mines when Haldane was old enough by tying a rope around his ankle and sending a young JBS down into the mine until he passed out from bad air. That way he'd know the air was bad, he would pull him back out again. And I think that probably had psychological consequences that may have explained some of his quirks. So the original conversation between Haldane and Medawar had to do with why Huntington's disease was so common. Now, Huntington's disease, as you all know, is a fatal neurological disease. It's an autosomal dominant disease. If you have the gene, you're going to get the disease, and the disease is going to kill you. And so the idea is, why does a lethal gene like that persist in populations at a fairly high rate? About 1 out of 15,000 people of Northern European descent have that gene, which means that that gene is going to kill them. What Haldane pointed out was that it probably has to do with the age at which the effects of that gene are actually manifest. So this is the age of onset of symptoms of Huntington's disease. What Haldane noticed is that most of the people who get Huntington's disease get it from their 40s and older, by the time which they would have had most of their children. And to the extent that you've had most of your children, or all your children, by the time a fatal disease strikes you, then evolution, natural selection, basically does not see that gene. That's a neutral gene, because it has no impact on the number of young you have. Well, Medawar took that basic idea that there is no necessary evolutionary cost associated with a lethal gene unless it has its effect early in life, and he ran with it, and he developed this elaborate evolutionary theory of aging that we still think provides the best general answer to broad patterns of aging today. Now, what Bill Hamilton did was he actually just put equations to that. So, well, if that's the case, then we ought to be able to put equations like this. So here's the idea. Let's imagine you have a gene like let's say Huntington's disease, but you don't get it till you're 100 years old. Well, clearly it doesn't affect the number of children that you leave, so it's evolutionarily neutral. It's neither favored nor disfavored. Same thing is true of a gene that's beneficial. If it has its effect very late, it's not important. It doesn't affect your reproductive success. If you have it very early, of course, it's very important. If you get Huntington's disease when you're 18, then you're unlikely to ever leave any children. That gene will quickly disappear from the population and only recur at whatever the new mutation rate is. What Hamilton did was he sort of quantified the rate at which the power of natural selection to affect the fate of that gene waned with age. And here's just shows from 2 populations that he had data on the rate at which the effect of the gene. So the effect of the gene is big when you're small. It gradually disappears. And if the gene has its effect later on, has no impact on fitness. An important thing to note is what causes this rate of decline. What causes that rate of decline is the rate at which individuals die by causes that have nothing to do with aging, that may have to do with eating bad hamburgers, with getting hit by trucks, with extrinsic hazards. If extrinsic hazards are high, then the population will be mostly very young individuals because nobody will live very long. If the extrinsic hazard rate is low, then lots of people will live to a very age, so it becomes beneficial for nature now to start providing you with protective mechanisms against immune problems, against heart disease and all. That's the basic idea. Now, because this fall off in natural selection, it leads to aging, or it can lead to aging by a couple of ways. One, the one that Medawar favored, was that if genes like the Huntington's disease have no impact on reproduction or survival until late in age, then those genes can accumulate. There's not selection for those genes, but there's no selection against them. So over the millennia, over the generations, genes of that nature, if they exist, can accumulate in the genome because there's nothing to sort of make them go away. 50 years on, we have very little evidence that that's the case. The other type of process that could lead to aging because of this waning of natural selection is that it's possible that genes have a beneficial effect early in life, but their cumulative effect over time is detrimental. But because their beneficial effect happens when selection is strong, the detrimental effect happens later on when selection is weak. The natural selection can favor those genes, and those genes will actively spread throughout the genome. Now, that process is called antagonistic pleiotropy. Pleiotropy simply meaning the genes have multiple effects, antagonistically simply meaning they have different effects at different ages. Beneficial effects early, detrimental effects late. Now, for the longest time, people always they liked this idea. It was superficially satisfying. Things that are really good for you when you're young could, in the long run, be bad for you. The problem was that nobody could find such genes. Williams had an idea. Williams said this: let's imagine a gene that hastens the calcification of bones during development, and that's beneficial because you grow up strong and sturdy earlier. But let's imagine that same process continues and leads to the calcification in your arterial walls later on. Clearly, that would be detrimental. But because of the beneficial effect early in life, that gene would be favored. For the longest time, there were only these hypothetical examples. But now, thanks to the last 15 years of molecular biology, we have dozens and dozens and dozens of these genes that have exactly that effect that we know about. And one of the ways that so before I get to sort of those specific genes, let me just tell you how you can test this theory. There's a couple of ways you can test it. It makes some specific predictions. One of the predictions is that if you can take this waning of natural selection and slow it down or move it out, take it from here to here, that you would predict you'd be able to evolve longer life. And Michael Rose started doing this around 1980 with fruit flies. And all he did was take normal fruit fly reproduction and move it back in time. How did he do that? He threw out all the eggs that any flies laid until they were quite elderly. And he only let those eggs form the basis of the next selection. So you only got to leave eggs that were going to make the next generation if you lived an exceptional long time. And then he gradually moved that later and later and later, the prediction being that you would create longer and longer live flies. So delaying reproductions over many generations should lead to increased longevity and preserve physical function. Because even though I'm talking mostly about longevity, that's really a shorthand. We don't really care if people live longer if they stay healthy longer. It just turns out if they stay healthy longer, they're likely to live longer. So Rose was successful. Over the course of years, he doubled the lifespan of these flies. They had enhanced function in a whole variety of measures, and this now has been done in so many labs that it's become almost a standard procedure. It's almost not interesting enough to bother doing anymore. A second way to examine this theory is by a second kind of prediction. And the second kind of prediction is that safe environments, meaning those environments in which you're less likely to die from these extrinsic hazards, should over generations and generations reduce the rate at which natural selection falls off and basically do the same thing that Rose did with his delayed reproduction. And this is basically where I came into aging. I thought this was a neat idea, and I thought I knew how to test it. I was studying opossums in South America. I found out they aged incredibly rapidly. Here's an opossum. This is opossum number 9. Opossum number 9 has a cataract. This opossum 3 months earlier did not have any cataracts. I captured her. This had nothing to do with aging. I was doing something else, but I was capturing the opossums every few months. And I noticed that within 3 months, they'd get arthritis, they'd lose weight, they'd get parasites, they'd get cataracts. They fell apart incredibly, like the one-horse shay. So I thought this would be interesting to study aging of these animals in the wild. Now, why would they age so fast? They basically don't live any longer than a mouse. Why would that be? Well, I thought, thinking about this evolutionary theory, it's because they are so prone to environmental hazards. They get killed by every predator you can think of, even when there are no cars around. And so they are absolutely, about 80% of opossums, as it turns out, are killed by predators. So I thought about looking for an island that had opossums but didn't have predators. There it is, right there, Sapelo Island, Georgia. I spent 2 years kicking around the southeast, opossum heaven, as it were, looking for the right island. It had to have 3 things. It had to have opossums, because otherwise the study would be so short. It had to have a lack of predators, because I wanted to know that. And it had to have no bridge between the island and the mainland, because I didn't want opossums going back and forth and breaking down whatever local adaptation they had. Key point to know is that about 80% of opossums on the mainland are killed by predators. Sapelo Island didn't have any of those, and it had been separated from the mainland, I knew, for 4,000 to 5,000 years. So that's 4,000 to 5,000 generations, and I was betting if there was going to be an effect, that would be long enough. So I went out to Sapelo Island. I radio collared about 40 females. I radio collared about 40 others at a nuclear weapons facility on the mainland. Now, why a nuclear weapons facility? Because nobody is going to mess with your experiments at a nuclear weapons facility. It's got this enormous fence and a bunch of people carrying machine guns all over it. So it's a good place to do field experiments. Even though it got a little weird sometimes at night when I was going out looking for my opossums, and I'd see people in moon suits sweeping stuff up off the highway. Always made you wonder. What did I find out? If this is the age-specific mortality rate of the animals on the mainland, mortality increases with age, as you can see. On the island, not only was the mortality rate lower, but the rate of increase was slower. So this was all consistent with slower aging on the island. This wasn't sufficient by itself to convince me that they were aging more slowly, but I also noted that reproductive senescence, or the ability to rear young in a second reproductive year, was very poor on the mainland. On the mainland, only about 20 to 25% of the opossums even lived into a second year, and if they did, they had a high rate of sterility. Their young in the pouch grew quite slowly. On the island, none of that was true. On the island, many more of them lived into the second year, and when they did, there was no difference in any aspect of fertility. And third, this really ties in with Bob Kohn's, because my other measure of physiological aging was actually looking at tail tendon fiber aging in the opossum, basically looking at collagen aging. And the tail tendon fibers of the opossums age more slowly on the island. Some of you may have seen this PBS TV series on aging that sort of had me doing this to some of the opossums. And of course, when the TV cameras are there, you're a little nervous when you're doing this, so I actually hit a major vein when I was opening this up, and this blood welled out, and the cameraman passed out. He had the big close-up lens on. The funny thing is that this sort of experiment, 7 years later, somebody did the same experiment in the lab in fruit flies, came to the same result. And so this has got to be one of the first times in the history of biology where somebody discovered something in an opossum, had it confirmed 7 years later in a fruit fly. The other thing that Darwin pointed out is that nature, if you look at nature, nature has altered aging rate again and again and again and again. These are not laboratory mice. This is how you handle mice that are what I call real mice. That is, these are the mice that our laboratory mice are descendants from. I've spent the last few years collecting mice from all over the world trying to replicate my opossum finding in wild populations of mice. Because the problem is, once I knew I had opossums that aged more slowly on the island and the mainland, and I wanted to figure out the molecular mechanisms, there's no genetics for opossums. Especially, there was no genetics for opossums in the early 1990s, but there were for mice. So this is I am. The NIH, thank you very much, paid for me to spend a rough time on these tropical islands, scuba diving during the day, trapping mice at night. And it turns out that wild mice live longer than laboratory mice. We've actually shortened the lives of our laboratory mice. The islands that I happened to pick to get these from, one of them, they lived a little bit longer than a standard laboratory mice, but not as long as one of the really robust mice from Idaho, which was where my best mice came from. So another aspect of this whole Darwinian contribution is an appreciation of this ubiquity. So this idea about antagonistic pleiotropy, there are trade-offs. Yes, you can live longer, but you have to give something up. Otherwise, nature would have made you live longer anyway. We now know a tremendous number of antagonistically pleiotropic effects. So now that we've had dozens of genes that extend life, we've had multiple chemicals and dietary treatments that extend life and things from yeast to mice, we know that invariably you get a change in body size, you get sterility or subfertility. If you have a larva, it's likely to die at a higher rate, weakened bones and muscle strength, increased vulnerability to infectious disease. It's always something that's traded off. Now, my colleagues in molecular genetics have always hated this, because what they're after is to find a way to extend life and have nothing else, no other effects that change life, that except for longer, healthier life. And this is a celebrity organism. This is C. elegans. You can see they wrinkle with age too. This is a young one. This is an old one. And there was a particular mutation called DAF2, which is a mutation in the insulin receptor that made these guys live about twice as long. Now, the interesting thing about DAF2 was not just that it made the worms live twice as long, but that it had none of these side effects, that it was basically a cost-free extension of life. Or so the worm biologists said. They said there's no difference in reproductive rate, they develop just as fast, there's no cost. And so for 10 years, I pestered them. I said, do me a simple experiment, would you? Would you take a group of these DAF2 mutants and put them in the same dish with a group of normal worms, and then just see who's left after a few years? And Gordon Lithgow, a good colleague at the Buck Institute, finally did this, and this is what he found. When he did that and he put in half of the worms were DAF2 and half of the worms were normal wild type, what happened? Well, experiment after experiment after experiment, pretty soon all the DAF2 animals were gone. Why were they gone? Well, it turns out there was a tiny difference in the reproductive rate early in life. Not enough to really bother the worm biologist, but enough to bother an evolutionary biologist. It turns out that we don't know of any genetic mutants, and like I say, we have now over 100 that extend life that don't have some sort of early life cost. Darwin also helped explain, and this theory also helped explain, a large and well-known but puzzling pattern. This is the pattern. Everybody probably knows this. Small mammals are shorter lived than big mammals, on average. And this was first discovered in the early 20th century, first sample of 6 mammals. And then in the mid-century, George Sacher did 63 animals and found this same basic pattern. And a number of people subsequently went on, and I came in a number of years later with 600 species of mammals, which we had good longevity data. And there's this is what it looks like. There's this good relationship. It's loose, but it's very robust between body size. If you're a small mammal, you live shorter. If you're a big mammal, you live longer, for the most part. Now, what's the mechanism? What causes that? Well, people speculated it's metabolism. Small animals have a fast metabolic rate. Large animals have a slow metabolic rate. That was a very popular theory for a while, particularly after free radicals were discovered, because oxygen radicals are an inevitable byproduct of oxidative metabolism. And so it certainly made a lot of sense. It turns out it really doesn't make sense if you look at it, because you actually measure oxygen consumption by these guys. It turns out that small animals consume substantially more oxygen per cell per lifetime than large animals do, even if they're short-lived. But from an ecological standpoint, it turns out that if you explore the variation within that relationship, it turns out that organisms or species that, because of anatomy, like this porcupine that seriously discourages predators, physiology, like bats that can escape bad conditions by simply flying away from it, or like naked mole rats that live underground in large colonies with extremely constant climatic conditions. All of these things are associated with long life, not in the wild, but long life even in captivity, where they're protected for all of those. Just to give you an example, this is same data, all the mammals, all these white circles, those are all the bats. How long do bats live? Bats live, for a 7-gram bat, they live up to 41 years in the wild. Some of the bats that you guys see flying around here in the summer could easily be in their 30s. Now, if you think about that, they not just have lived 3 or 4 decades, but a bat in the wild has to have really good high-frequency hearing, because that's how they hunt, right? How good is human high-frequency hearing after 30 years? Well, I know my kids have a cell phone ring that they use when they don't want their teacher knowing it, because its frequency is higher than the teacher can hear. Your hearing frequency starts to decline when you're still a teenager. So they've maintained that high-frequency hearing. They've maintained the speed and ability to catch an insect every 3 to 5 seconds to keep from starving to death. They fly 50 to 100 miles a night, and yet they find their way back to their roost the next morning. So they've preserved spatial, learning and memory, hearing, muscle strength and endurance for a long, long time. They live, in fact, about almost 10 times as long as an average mammal of that size should do. The final contribution of Darwin has to do with what I call phylogenetic thinking. Here's a famous entry out of his notebooks from 1837, where he first conducted the first branching diagram of how he saw life evolving. Species splitting apart into new species, those splitting apart again. This is really a way to represent evolutionary history. This is what it looks like if you actually do this from the molecular systematics of mammals. Before 2001, we didn't know this. We couldn't put this up there. There wasn't enough information to really determine what the relationships, the evolutionary relationships of all those mammalian orders were. Now that we've done this, what we can see is that long life, and by that I mean animals that live at least twice as long as they should for their body size, has evolved over and over and over and over throughout the mammals. Mammals, long life, short life, long life, short life, all the time. There's no problem for nature to create animals that are longer and longer and longer life. There should be no problem for us to figure out how they do it so that we can make the 150-year-old human. Some work that I'm doing, we're taking a smorgasbord of animals that are related to one another evolutionarily, but differ dramatically in longevity, and look at the molecular mechanisms that underlie that. And I don't have time to talk about today. If you invite me back, I'll talk about what we're finding from that sort of study. That's just what the species are. This is how long they live. So we have a mouse versus a naked mole rat, which they're not just cute, they also live a long time. So why am I confident that we will live 150 years and sooner rather than later? So methods for extending life in worms, alteration of more than 150 genes has now been shown to extend life enough that if a worm were a human, then Jean Calment would be a 150-year-old human. We also can extend life by restricting food, by sensory, some sorts of modulation of the sensory apparatus, by certain diets, by certain drugs. I'm not in love with any one of these as a necessary extension to humans, but we have so many ways of extending life, it's very difficult for me to imagine that some of them aren't going to be applicable. That's not just true of worms, it's also true of mice. We have more than 20 genes that extend life long enough to make a mouse into the mouse equivalent of a 150-year-old. We can restrict methionine, we can exercise mice and make them. We have lots and lots of chemicals that now extend life. I just wanted to mention one, which is the NIH-sponsored intervention testing program, which take a drug. If you have a drug you think will make mouse live longer, then they'll test it for you. There are 3 places that do it simultaneously, so the results have instant credibility because the same experiment's done at 3 sites simultaneously. Let me just show you the most surprising result yet. This is the drug I want you to focus on is called rapamycin. Rapamycin basically damps down the TOR pathway. It's currently used in transplant surgery to prevent organ rejection. They actually didn't start this experiment because of technical difficulties till the mice were 20 months old. Now for a mouse, that's about equivalent of a 50-year-old human. So they started giving the mice this drug at 20 months of age, which I don't even know why they bothered because no one would have expected it to have any effect. But look at it actually had a substantial effect on lifespan even when started at 20 months of age, even in both sexes, which quite often there are sex-specific effects. So it's this kind of thing. I'm not saying rapamycin is the key to make us live 150 years old, but there are so many things that we have going. Some of it's going to work out. Let me just finish up with something that you don't often get in sort of scientific talks, which is a little bit about ethical issues. So I've done it. I've invented longicin. It's going to make most of us live to be 150 years old. If it halves the aging rate, if you start it when you're 40, by the time you're 100, you'll only look like you're 70, and it's cheap. So the question becomes, well, that's great. Now do we want to give it out to everybody? Now, to me, that seems such like an obvious no-brainer that this would be the greatest thing since the opposable thumb, because remember, if you slow aging, you not only retard the progression, delay the onset of all of these fatal problems, but there's all of these other things that can make life less enjoyable than it does. So this is better than curing cancer, better than curing heart disease, because you're doing both at once, plus you're dealing with joint pain, sleep disruption, sensory deficits, all kinds of things. So this is fantastic, you know, and New Yorker cartoonists thought it would be fantastic as well. So I was quite surprised when I got a phone call from Leon Kass, who was the head of the President's Council on Bioethics at the time, that asked me to come to Washington and sort of talk about the ethical implications of this research. To me, I had never thought before that everybody wouldn't think this was the greatest thing. I originally wasn't going to go, but he was so nice and persistent, and he invited me and Jay Olshansky, so I thought, oh, we'll do our little wager dance in front of him. So after they determined that embryonic stem cell research was no good, they wanted to look at some other things. So as he said, they wanted to provide an ethical lens through which to view particular developments in their proper scope and depth. Now, after I did that, it occurred to me, gee, the problem is I only talk to other people that are biologists. And when I do that, they all think this is great because they're all biologists working on this. So when I gave public talks, I used to say, okay, we've got this thing, you can live 150 years, it prevents all these diseases, you're healthy. How many people think this is a good idea? And when I did that, I would find out that 80% of the people in the room thought that's a really bad idea. It was kind of disappointing to me since I've sort of staked my professional career on investigating this. So I think it's worth, actually, let me get it. So how many people think that's a good idea? How many people think it's a bad idea? Well, this is better than most audiences. This looks like it's sort of 70-30. Let's see if we can make it 80-20 at least. Why are people ambivalent about this? Because it's not rare. And again, I'm usually talking to audiences who've come to hear a lecture on aging. So you'd think they'd be predisposed to like this idea. So let me go through 4 things that I think should be taken seriously. The first one is the Tiffanys fallacy. Now Tiffanys, well, these are the arguments, the naturalistic argument. So Tiffanys fallacy, what we're going to do is we're just going to keep people alive who are frailer and frailer and frailer, and nobody should want that. The naturalistic argument is, look, people are supposed to live 70 or 80 years, and then they're supposed to get sick and feeble and frail and die. That's the way, you know, God meant it to be, and that's the way it ought to be. There's fairness issues about, well, first of all, how much is this going to cost? And third, if it's going to cost a lot, is it going to be available to everybody? I try to get around that when I do this by saying, look, this is really cheap. It's like aspirin. But we don't know if it will be or not. And then the final thing is sort of Malthusian issues, which seem to be the biggest one. Well, look, there's going to be, you know, we've got enough people anyway. You know, we're running out of natural resources. There aren't enough tenure slots for new graduate students. You know, there's all kinds of resource shortages to deal with. So the Tiffanys issue, I think, is easily dealt with because we now know a lot about what animals and people look like when they stay healthier longer. It's not that they go through this extended period of feebleness and frailty and disability at the end. Just to take an example, if you take a calorically restricted rat that lives longer and stays healthier, and you put a running wheel in, and you look at how much they run, this is what a normal rat does. By the time they're 18 months old, they've pretty much quit running at all. But look at these calorically restricted ones. They're still running a kilometer a night when all of the other, you know, well-fed rats are already dead. So the same is for people. These healthy centenarians, when they go in a hurry. By the time they get sick, which is usually the first time they go to a doctor when they're 103, you know, then they're not sticking around long after that. So that's empirical evidence that that's not a problem. What about people in a state of nature? I feel particularly close to this because I've worked with people in Papua New Guinea who were living reasonably close to a state of nature, and it's not a great life. We have a few survival curves from societies in a state of nature, and as you can see, the life expectancies are around 20 years. There are many diseases. Most people are dead before they're 40 or 50. There are some that live later, but it's it's Hobbesian. Nasty, solitary, brutish, and short. And the other thing is, it's gone. Our natural lifespan, it's long gone. Good riddance. Finally, penultimate, there's the fairness issue. Is this going to be available to everybody? Well, I don't know. David Sinclair, who's just sold his company for $720 million, thinks it's going to be cheap. But if I just made $720 million, I might think it's going to be cheap too. Or no matter what the cost is, it would be cheap to me. Well, my response, yes, it may or may not be. It may not be cheap initially. But my response is, we don't care about that. Societally, we don't care about that. It's demonstrated already that we don't care about that because we don't not give people CAT scans and MRIs and anti-HIV drugs because we can't give it to everybody. Most philosophers will say if you can do something that's good for some people without making things worse for other people, then that's an objective good. We don't not do heart transplants because we can't give heart transplants to everyone that need them. So I think the fairness issue has sort of been settled societally already. But there's the resource issue. And the resource issue is a very serious one, and I wouldn't try to say otherwise. That is, we have 6.3 billion people already. If we slowed the rate of aging, the population would grow faster. It wouldn't grow as much faster probably as your intuition tells you, but it certainly would grow faster. And my response to that is that's true, and it's something to be concerned about. However, we're also going to have to be concerned about it whether we do anything about aging or not, because the population is still tending to increase, and we're going to have to find ways to deal with that. Now, that kind of attitude, the burden of supporting old people is too heavy, politicians are disturbed by the enormous cost of supporting the aged, costs of maintaining these people become even heavier if they live longer. Funny thing about this, it was said by Elie Metchnikoff in 1907 when he was living in Paris. The life expectancy at that point was 50 years. Somehow, the French have muddled on, supporting the unbearable burden of all those old people for another century, and somehow have managed to endure. So I'm sure if we think about it and we plan and we use our human ingenuity. Oops. Let me just finish up here. Is that if we're really serious about dealing with research, we do lots of things that typically we don't do. So let me just finish up. The rationale for slowing human aging, the thing that a number of us are working towards, is that it's not really the prevention of death that we want to extend healthy life. I would love to be able to have a conversation with Rudyard Kipling. So we want to improve health and we want to extend health. That's the same goal of all disease-based biomedical research. I have never said, how many people think we ought to try to cure heart disease and not had everybody raise their hand? How many people think we ought to cure cancer? Okay, everybody raise their hand. How many people think we ought to do both at the same time? Oops, 80% of the people think that's a bad idea. So this really is a similar approach to curing diseases. It's just a more effective approach. If we continue to cure diseases or treat them better, but we don't do something about aging, then we're going to have many, many more disabled people with dementia, and that's not good. So I think I'll end there. I think I've run over my time a little, and see if there are any questions. Thanks for coming. Thanks for your attention.
We have time for a few questions. microphone, please. We're going to pass the microphone because the answers are taped. So could you please? Dr. Matsuyama.
Well, thank you very much for a very exciting talk. So you mentioned basically about aging and lifespan of animals. So, but when we look at the world from the viewpoint of evolutionary theory, the plants, there are some cedar and pines, they are living 1,000 years. And then do you think that do they have a similar lifespan program, or they are completely different organisms?
Yeah, so interesting question about aging in plants, in vegetation. It's a very different phenomenon. I usually don't talk about it because I think it's hard to make these two things equivalent. So plants are what we call modular organisms. That is, they produce new modules. You know, you can take a pine tree and you can, you know, plant pieces of it and grow up lots of new pine trees. Also, a lot of plants are mostly dead. Like the oldest bristlecone pines, one of the oldest trees, if you look at them, what they've got is this very tiny, narrow range of cambium with a couple of leaves that are still alive, but mostly all the organism is dead. To me, that's enough different that I don't know how to think about it in the same way that I have to think about us, where we can't have our arm die, but the rest of us, you know, is good, or an arm and a leg go, you know, and only our eyelids work, but we're still alive. So it's really interesting, but I think it's a different problem, and I don't know how to sort of make it consistent with the way I think of aging in in animals, most animals.
Fascinating talk. The sequence of molecular and cellular changes that bring us through embryology and into maturity continue as a sequence of change throughout our lifespan and contribute to aspects of aging because of this changing environment. If you're going to increase this time span, aren't you going to be affecting this sequence of change, which actually keeps us, I think, alive and keeps us biologically fit? I mean, it's part of your thesis. If you change the front end of the Darwinian curve, you're going to affect the back end, or if you affect the back end, are you going to affect our front end?
That's a good point. And that's why I think it's important to look at these results where we've started some sort of therapy, whether it's reducing diet or giving drugs, in adulthood. I mean, I think you're right. Certainly, I think aging is separate from development. I think it's different, but it doesn't mean it's not affected by development. So the way I think of this from an evolutionary viewpoint is I think of this symphony of gene action that's exquisitely designed to produce a healthy young adult. So there's a conductor. Natural selection has orchestrated this. But what happens is when we get to be adults and we start getting later into reproduction, it's like suddenly the conductor walked off stage. There's nothing orchestrated anymore. Natural selection has not orchestrated that, and so pretty soon the violins start to go off in their direction. And so I actually think that an important part of it is probably some of those developmental programs that go awry, the genes that continue on when we'd be better off if they shut off, or they shut off when we'd be better off if they continue. So I think we now know enough about the hierarchical structure of the genome to know that there are lots of genes that if we just tweak with them, we seem to be able to preserve this nice symphony for a bit longer. I don't think we're ever going to get immortality. I think that's asking too much. Even E. coli, even bacteria age, even though we never used to think they are. So I don't think it's an insuperable problem. I think the problem of immortality is an insuperable one. But increasing human health another 20 to 30%, well, there's so many ways we can already do that in experimental animals. It's hard to imagine some of those aren't going to have relevance. But I do think there's going to be side effects. I mean, this is part of the antagonistic pleasure. The nature of those side effects is going to determine whether it's worth it. Let's imagine, for instance, one of the side effects that we're just discovering about this food restriction, which we've known for 70 years, is that certain aspects of the immune system do not do well when you restrict food. You know, that's not a surprise to immunologists, but it's a surprise to a lot of biogerontologists. Could be that one of the side effects that you're going to have to deal with is some sort of immune compromise. So that if you're going to get knocked off by the next flu epidemic, then this might not be a good trade-off. So I think trade-offs are ubiquitous. We're not going to be able to avoid them. The question is, are there going to be trade-offs that we can live with or not? That's a long-winded answer. I hope I at least got at a little bit of your question.
So, Steve, the question is, out of a sample size of more than 7 billion human beings, how come we don't have more of those super centenarians? Is it because we're not sufficiently outbred, or we're not sufficiently shielded in our stressful lives?
Well, I mean, she was a real outlier. I have to say, Jean Calment was a real outlier. So since she died in 1997, nobody has come within 3 years of living that long, even though we now have over 1,000 people that we know have lived to at least 110. I think our physiology is buffered to the extent that single gene changes don't have these kinds of big effects on us. That's why when I actually think about how this is going to work out, I actually think we're going to find something that, you know, one of the big problems with aging is chronic inflammation. It tends to increase. I think we're going to find ways to make that a little better, and something else to make our mitochondria work a little better, and something else to do this. I actually think we're going to have these really small incremental things. The problem is we're not going to know if they're working until, 50 years. People go, geez, those people that have been taking those statins, they're really looking good, Do you believe that person's 90 years old? She's been on statins since she was 30. It's going to happen like that, I think.
One final question, Dr. Lamanna, and then I would like to invite you to join us for the reception, and please keep asking questions directly with Dr. Austad. Thank you.
I wonder whether, do hibernators have an extended lifespan, and if so, do they extend it beyond the time they spend dormant? Is that a strategy for extended lifespan?
Some of them do. So if you look at the bats, and you say, well, does this bat species, which hibernates 8 months a year, live longer than this one that hibernates 6 months a year, live longer than this one that never hibernates at all? There's a very small impact of hibernation, but it's not as much as you would think. It's not, being a bat, not even hibernating, is much more important than being a hibernating bat. There is an effect there, but it's a smaller effect than you would think. All right, thanks very much.
Dr.